When we are doing this interview right now, both of us are using something like 5 watts. What is 5 watts? It's a flashlight. It's the power for a simple flashlight.
Professor Jack Tuszynski was born in Poland, where he graduated with a master's in theoretical physics. He obtained his PhD in condensed matter physics in 1983, and since 1993 has been a full professor in the Department of Physics at the University of Alberta.
Dr. Tuszynski is now at the cutting edge of research about the biophysics of cancer. He has published over 600 journal papers and 12 books. Jack Tuszynski.
And then people say, yeah, but computers are more powerful. They can win at chess with humans. Blue gene computer beat Kasparov at chess. Well, wait a minute. This blue gene is powered by 1.5 megawatts of energy, which is basically the needs for a small town, 30,000 people for their households. We experimented with measuring the metabolic rate, consumption of nutrients and production of ATP through mitochondria. And we did this on cancer cells exposed for about 10 minutes to near infrared radiation. And we saw something similar, 3 to 5-fold increase in the energy production when cells are exposed to electromagnetic fields.
As a final comment, you can give the audience, because you're a cancer researcher, your current hypothesis about what's happening with cancer.
Welcome back to The Boundless Body. So check this out. 63.5% of you who are regular listeners haven't subscribed yet to the show. So please take a moment. It would be really helpful to click subscribe. It's free of charge. It will really help us out. In turn, we'll help you out by bringing you better and better content. Along those lines, send us an email with your feedback, boundlessbodypod@gmail.com. Send us your feedback about how you think we can do better. Give us questions to address on the show, interviewees you'd like us to interview. It'd be super helpful. Okay. Let's be a community. So today is amazing. We have Jack Tuszynski. He's a biophysicist, and he really helps to walk us through the bioenergy of the cells that are living in our bodies. And it's not woogie stuff. What it is it's hardcore bioenergetics. It's the bioenergy of the body. Energy is talked a lot about in the healing arts and even in energy psychology, but Jack walks us through a really grounded, concrete way of understanding the potential for quantum computing in the body via calcium ions. I'm all about calcium ions. Michael Levin talks about calcium ions quite a bit. I talk about calcium ions a lot in my upcoming Visionary Somatics book. And Jack walks us through why he thinks it's the best contender for quantum computing in the brain. We talk about cancer. We talk about mitochondria. It's an awesome discussion. Please enjoy the show. Have a great one. Okay, Jack, welcome to The Boundless Body. Really excited to have a biophysicist on board.
My pleasure. Thank you for inviting me.
All right. So excited to talk about calcium ions and quantum physics and microtubules and fascia. I'm just really geared up for the conversation. I'm excited.
Lots of topics to talk about, for sure.
Yeah. Okay. So going to start out with a bio. It's a longer bio than usual, but it just the content is so rich. So I just want folks to know a little bit about your history. So Professor Jack Tuszynski was born in Poland, where he graduated with a master's in theoretical physics. He obtained his PhD in condensed matter physics in 1983, and since 1993 has been a full professor in the Department of Physics at the University of Alberta. Dr. Tuszynski is now at the cutting edge of research about the biophysics of cancer, and has produced a drug that is currently in clinical trials to treat aggressive bladder cancer. He has published over 600 journal papers and 12 books, and is on the editorial board of 30 international journals. For real, 30 international, that's a lot. His research interests are strongly linked to the protein tubulin and the microtubules assembled from it. We'll go deep into tubulin and so on. So due to its prominent role played in cell division, tubulin is an important target for anti-cancer treatments. More recently, Dr. Tuszynski has focused his research efforts on the fundamental issues related to the role of quantum and electromagnetic effects on living systems and their higher functions, including memory formation and consciousness. All right, Jack. You've been a busy man.
Indeed, indeed.
Yeah, and you're an interdisciplinary researcher, and that's what I just love. I'm an interdisciplinary guy. If we want to figure out life in its complexity, we got to come at it from different angles. Yeah.
Indeed, that's the future of science. I think we have too many silos, and we need to change it.
Yeah, for sure. So I was thinking that we could start out with just a basic description of the cell. So, one of the significant aspects of your work is, of course, your research with tubulin and microtubules, but this is going to be new to a lot of listeners. So if you could just talk about the structural scaffolding of the cell as a kind of foundation for folks first.
Right. So it could be, I would say, a cartoon representation of a cell, because we can go really deep into the weeds, but we don't want to do that, I guess.
Let's start with a cartoon. Good idea.
Right. So for me, as a physicist, the way I see a living cell is something which is, well, has different morphologies, different shapes. But let's say the main thing to begin with has boundaries, which is called a membrane. So the membrane is composed of phospholipids and some ion channels inside, which we'll come back to, because it allows for the transport of material in and out of the cell, and the communication with other cells, sometimes um, uh, creating structures together with other cells for forming uh, tissue, basically. Okay, multicellular organisms. But for a single cell, and again, dimensions matter. So when I teach biophysics, I usually talk about something like spanning the range of from 1 micrometer across for bacterial cells to as long as a meter for some giant squid axons, neurons, and even in the human body, right? Some of them are as long as a meter. So 6 orders of magnitude in terms of the dimensions. And then you go inside, look inside the cell, and you see, of course, the nucleus. In eukaryotic cells, such as our cells, all cells will have a nucleus with DNA inside, packaged into, with other proteins. DNA is packaged and folded and so on. Then we can later on come back to what happens when the cells divide. In addition to it, outside the nucleus and between the nucleus and the membrane, there's something called the cytosol. It's basically fluid, but that's again not quite true to say it's just a fluid, because it contains a lot of structures organized, and even water, which is like, let's say 70% of the total mass of each cell, is not quite your running water from the tap. It is structurally oriented, typically around surfaces such as the surface of proteins. So inside you have proteins and some other biomolecules and ions. But proteins would be the largest structures that make up the cytoskeleton, which is basically structurally stabilizing the cell. And sometimes participating in processes such as cell movement, motility, cell division, and transport. So within this cytoskeleton, we have 3 main organized protein structures: actin filaments or microfilaments, which are the smallest of the 3, and they are typically organized close to or localized close to the membrane, so on the kind of periphery. It's cell cortex, and then you have intermediate filaments, which are a bit more randomly organized to provide sometimes more structural integrity, and then microtubules, which is the structure of great interest to me and has been for decades. And microtubules are made up of the protein called tubulin, and they form kind of cylindrical tubes. And they are special for various reasons, including the size, which are the largest of the three types of filaments, and also very highly charged electrically, which means that they will be interacting with or strongly dependent on ionic concentrations, pH, and interacting with electromagnetic fields. So and then you have organelles, which I will just mention the importance of mitochondria, because the cell is in a way, it's dynamic. It's very dynamic and requires energy, biological energy production, or actual transduction, because we can't produce energy. We can only transform one form of energy into another. In this case, mitochondria transform the energy contained in glucose or similar molecules into what we call sometimes the quantum of biological energy, namely ATP molecules. And this is the currency of energy in biology, by and large, ATP and GTP. For everything I say, there'll be an exception. So please don't jump on me, because I want to be very brief here. But ATP carries what I call a quantum of biological energy, and this quantum is used for many processes. And contrary to some misconceptions, most of the processes that take place in the cell are organized and directed, not random and diffusive. There is diffusion too, small molecules like caffeine, which I just had a cup of coffee before we started. They diffuse, but important things like transport of molecules or vesicles or movements, cell movement or cell division, they are all organized and orchestrated, and they demand some input of energy. Every step, including, let's say, transcription and translation of DNA, every step of the process requires at least one molecule of ATP. So that's why we call it quantum of biological energy.
Yeah.
So that's kind of a very brief summary how I see it as a physicist, and I'm sure biologists will correct me with everything I said, but
Well, I mean, there's so much complexity that you got to have ways of kind of cleaving down and reducing, and I think that's a great reduction. So, is it true that mitochondria emit photons?
okay, so that's a big topic. But I would say the following. So mitochondria, let's start by saying what they do and how they do it. So as I mentioned, they produce in a very long chain of processes chemical reactions to begin with. So it's glycolysis precedes oxidative phosphorylation. There are chains of reactions leading up to the energy production. But in the process of energy production, mitochondria require a transfer of protons, so H plus ions, across the inner mitochondrial membrane, and simultaneously a transfer of electrons, which is, by the way, a quantum mechanical process, hopping or tunneling, using the language of quantum physics, that are coupled together. And at the end of the day, from one molecule of ADP, you produce a molecule of ATP. ATP is high energy molecule, ADP is low energy. So you add one phosphate group in the process. And because of the electrostatic repulsion between the negatively charged phosphate groups, you store this. It's a bit like a small battery, molecular battery, if you will. So you recharge this molecular battery, and that can be used for subsequent processes. It's the bottom line is that one molecule of glucose produces on average about between 32 and 36 molecules of ATP. And each of these molecules of ATP can be used for example, very importantly, molecular transport in cells involves building roadways such as actin filaments and microtubules, which I mentioned earlier, on which little vehicles move called motor proteins. So such as kinesin, dynein on microtubules, they move in certain directions. So you have organized freeways basically with, the directions of traffic.
It's towards the negatively charged end?
Well, it's both. So
It's both. Ah, okay.
both directions. But the beauty of it is that there are different molecules moving in one direction and different molecules in the opposite direction. So kinesins move on microtubules to the plus end and dyneins in the opposite, the minus end. And for actin, you have myosins, the family of myosins. But what I want to say is that each of these molecules, these motors, little cars, if you wish, or transport trucks, um, for each step on that filament, because the filaments are granular, they don't have a continuum of structure. They have building blocks or bricks, if you will. So each brick is a protein, it's either tubulin for microtubules or actin for microfilaments. So for each step of that movement, the machine, the little molecular machine, uses one molecule of ATP. So you see, this movement is quantized and granular again. So it goes click, click, click. And if it doesn't receive a molecule of ATP, it will stop and eventually detach from the filament. So you can imagine these highways inside the cell and transportation. So forming transportation networks, and then you have the transport taking place with cargo attached to these little machines.
It could be, and many times it's a vesicle, right? Carrying, for example, a dopamine molecule or an or an enkephalin or something like that.
Exactly. And in fact, even mitochondria are transported within the cell from place to place by on microtubules, and they are being moved from place to place by kinesins.
Yes, and just to insert in here just a little bit from neuroscience that, during the early attachment relationship, first 3 years of life, the brain is like a controlled explosion. It's growing so quickly, and it's growing in a relational field. It's growing in an attachment relationship. So in that attachment relationship is either good or bad, and it has a lot to do with emotional regulation. It has a lot to do with managing the intensities of being an infant. And during these attachment relationships, the mitochondria migrate out, exactly how you say, via these microtubules out to the growing tips of the dendrites and neurites, and are literally the battery, as you said, the bioenergetics for growth during the attachment period.
Exactly. And so back to your question. So mitochondria, they move on microtubules, and they deliver energy because they produce energy where needed, where and when needed. And you asked about the biophotons. So this has been studied by several groups, but I think the most interesting results come from the work of Professor Günther Albrecht Bühler from Northwestern University. And he has this wonderful website called Cell Intelligence. If you Google Cell Intelligence, you should find that website, where he demonstrates from experiments and published many papers on this, that mitochondria are the most likely places from which cells emit photons called biophotons as a byproduct of this process of proton transfer across the inner mitochondrial membrane. So when charges move across electric fields, they emit electromagnetic energy. And I published some papers with my colleague John Dixon demonstrating the correlation with the wavelengths detected in Günther Albrecht Bühler's lab to these processes. And so that's one source of biophotons. That's probably not the only one. There are also documented studies of biophotons coming out of the recombination of free radicals, which is also by free radical production, reactive oxygen species and reactive nitrogen species are byproducts of metabolism. And then they recombine, and when two charges attract, oppositely charged molecules attract and recombine, then the energy difference is emitted in terms of the electromagnetic wave or photon.
Interesting. And a lot of those reactive oxygen species and so on are spinning out from mitochondria.
Right. Exactly. And there's the opposite process, which actually that's kind of an interesting side note, if you allow me to spend a minute on this or two. So two things. My colleague, Professor Aristide Dogario from the University of Central Florida in Orlando, published work showing that the emission of biophotons, he measured that. He's an expert in quantum optics and has very sensitive instrumentation. He measured the amount of biophotons, or the intensity of electromagnetic field emitted from cells, and demonstrated a 3-fold or even 5-fold increase when these cells are provided with nutrients. So in response to the glucose that mitochondria use, and the byproduct would be electromagnetic energy. So that increases dramatically as a result of feeding the cells with nutrients. That's one thing. And the other thing which we did in my laboratory here is kind of the flip side to it. We experimented with measuring the metabolic rate, or the consumption of nutrients and production of ATP through mitochondria. You can have a biochemical assays telling you how much of ATP was produced. And we did this on cancer cells, exposed for about 10 minutes to near infrared radiation. And we saw something similar, 3 to 5-fold increase in the energy production when cells are exposed to electromagnetic fields in this near infrared range, which is specifically about 810 nanometers. We used devices which are produced by a company from Toronto called V-Light. So it's not a paid advertisement, but I'm just giving you the details. And so we tried different exposure times, 1 minute, 10 minutes, 15, 20, 25. And it's interesting because the increase in the metabolic activity of these cells is not linear. So it appears that 10 minutes is the best, optimal. So you feed them, expose them to this infrared light from the device for 10 minutes, and they respond optimally. So what does it tell us? Well, this device actually is used therapeutically. It's commercially available, by the way, and it's used therapeutically for dealing with depression, or even bipolar disorder, some neurological diseases, TBI, traumatic brain injury. By the way, I had a concussion about 2 years ago, and while on vacation in Tuscany in Italy, and I got a device like that from a colleague of mine with whom I collaborate. They have a company in Philadelphia called Quiet Mind. We haven't really experimented with their product, but it's kind of similar principle, and I used it for about 4 weeks, and I was back to normal. You wear it for about 10 minutes a day on your head in a helmet with this near infrared electromagnetic field being produced, and I felt better. So it's just anecdotal, but
I mean, so you treat mitochondria effectively, you dramatically impact the health of the organism, because mitochondria are the fundamental source of bioenergy. Like in my field, there's a lot of woogie energy talk, But when it comes to mitochondria, this is not woogie energy talk. This is bioenergetics.
Exactly. If you can deliver energy directly to the cells, like through this near infrared radiation, it's, by the way, non-ionizing and non-thermal by and large. I mean, over that period of time, anyway. So the energy, we at least believe that the energy is delivered resonantly, not by heating or not by forming, ionized solutions or something like that. So you energize the cell. We don't know exactly, what particular aspect of this, but I'll come back to it. Maybe we know a little bit at some glimpses.
Yeah.
But at the end of the day, the cell must absorb this energy and use it. So in the case of neurons, you probably know that there's this seminal work of Professor Magistretti from Geneva, I think, Switzerland, anyway, Lausanne or Geneva. I think he worked in both universities of Lausanne and Geneva. He demonstrated that glial cells in the brain provide the needed energy for neurons, because neurons typically are not sufficient in energy production. Neurons predominantly use mitochondrial oxidative phosphorylation, the longer, more efficient process, but that's not enough. And glia cell use glycolysis, which is shorter. It's a strictly chemical set of reactions, but more rapid. Just like when you exercise, run a marathon, you have lactic acidosis, you have lactic acid buildup in the muscles because of the glycolysis, which lactic acid is a byproduct of this. So that's the faster process of energy production than oxidative phosphorylation mitochondria. It doesn't require mitochondria. So what I'm saying now is that when you deliver this particular form of electromagnetic energy, you may provide the missing amount to neurons, and therefore the neurons then can perform functions which otherwise would be difficult, whether it's information processing, cognitive, or some other. In this case of traumatic brain injury, by the way, it's demonstrated that the microtubules get broken into pieces, neuronal microtubules.
Yes, and that glial cells are radically implicated in TBI because there's this thing called microglial tears in it, and it's probably relating to the tubulin and the microtubules of the glial cells. Yeah,
Exactly.
which spins the brain's metabolism into inflammation, because the glial cells are kind of like the bridge between the nervous system and the immune system.
Exactly.
Yeah.
So what I'm trying to say, hypothesize, I guess, is that when you deliver this electromagnetic energy to the brain, this energy can be used to repair the damage done by TBI, mechanical damage to microtubules, restore the connections, build new missing tubulin building blocks of microtubules and maps, microtubule associated proteins called maps for short. They interconnect microtubules in neurons especially. So what I talked about in the beginning about cell architecture, there is an important difference between dividing cells and non-dividing cells. So dividing cells, most of them, and non-dividing would be neurons, for example. In neurons, microtubules are oriented parallel to the axons and dendrites, form parallel bundles, which is not the case with dividing cells, because in dividing cells, you need to create an aster, a spherically spanning group of microtubules that will then divide the cell, chromosomes, and so on. But coming back to neurons, so these parallel bundles are stabilized by scaffolds formed by microtubule-associated proteins, which basically do something like this. So my hands are microtubules, the orange ball pen is a map, microtubule protein, and it's basically staples them together. And there's thousands of these maps stabilized. Now, when you have a brain injury, traumatic TBI or some other concussion or something like this, then these scaffolds may basically fall off, and the microtubules may break into pieces, and then you have structural problems inside. And by the way, that's also what's happening with Alzheimer's. In Alzheimer's disease, these maps, especially a special type of map called map tau, spontaneously detaches from microtubules. There is no mechanical damage in Alzheimer's by and large, right? I mean, most Alzheimer's patients are not suffering from the disease because of a TBI or something. It's basically the origin, I think it's debated, what is really the origin, but the consequence of it is known. And one of the two aspects is the neurofibrillary tangle formation, which means these taus, these red ball pens, form clumps. They aggregate instead of performing this important function of interconnecting microtubules, and that weakens microtubules. And in the end, over years, I guess, typically, neurons die down. They are not stabilized. Microtubules are gone from inside the axons, and that will lead us probably in a few minutes to another function of microtubules in neurons, which we hypothesize is memory formation. And that would be a very good link to Alzheimer's.
Yeah.
So we have the microtubules in neurons, we have the TBI, we have the neurodegenerative disorders, Parkinson's, Alzheimer's, and Huntington's, and all that stuff. In all of which microtubules or motor proteins in microtubules are dysfunctional in some way, and there is a pathology due to various reasons. But just to finish this thought and bring it back to the electromagnetic energy production. So now this company V-Light is claiming that you can help Alzheimer's patients by exposure to these electromagnetic fields, which might have a mechanistic explanation, because if you provide the energy that is not there, not enough anyway, then the cells may rebuild these connections, or at least prevent the further deterioration of the cytoskeleton. Okay, so that's on that.
Brilliant, because mitochondria are implicated in so many mental health issues, so many neurodegenerative disorders. Mitochondrial DNA is distinct from nuclear DNA, right? And mitochondria replicate much faster. Yeah?
Exactly.
Yeah.
Exactly.
So if you get a mutation in mitochondria, you're going to get this rapid mutation thing. And, when you have long-term stress, when the brain is stressed long-term, cortisol is going to go way up. The whole system is going to get desensitized to cortisol, so that the anti-inflammatory effects of cortisol won't kick in, and we have inflammation. With inflammation, we have more reactive oxygen species. The brain's metabolism spins into inflammation, and then what do we have? We have more mutation in mitochondria. We have more likelihood that the cellular architecture, the microtubules, are going to break down, and then the blood-brain barrier can start breaking down, which is another major problem. Yeah,
exactly, and it's related to Alzheimer's, for example, right?
Yeah, for sure. So I think that's just wonderful, kind of like figuring out a way with these electromagnetic fields to feed the mitochondria and kind of restore them. That's the hypothesis.
That's exactly the idea. We are working on it. I think we'll be doing some experiments soon on neurons. I haven't really worked with neurons yet, but we've worked with other cells. But maybe if you allow me to say a few words about energy, because it's important to understand. And people will say, "Oh yeah, that what are you talking about in terms of energy?" So we've done some calculations on the energy, and it's amazing actually, because um, the energy demands of the human body for its normal functioning are, we talk about calories, right? So we talk about 2,000 calories a day, right? Something like that. Maybe 1,600 for women, and big athletes maybe 3,000, but on average about 2,000. And if you do the math, it turns out that this corresponds to, in physical terms, 100 watts. Maybe most of your audience would not immediately relate this number to anything, but I can tell you that that's roughly the amount of power that is needed for a typical incandescent lamp. So that's 100 joules per second. That's what the entire human body requires for all of its needs. 25% of that is used by the brain. Brain is only about 1.6 kilograms. So that would be 3 pounds, maybe 4 pounds, something like that, three and a bit, which corresponds to about 2% of the body mass. So 2% of human body mass is equivalent to the brain, to the mass of the brain or weight of the brain, and consumes 25% of the energy, which is 10 times the average. So the brain, while consuming a lot of energy, and most of it, by the way, is used for things like maintaining the temperature, repairing proteins, and repairing damage and stuff. So let's say if we so it's 25 watts. Let's stay with this number, 25 watts. But I would say less than 10% is used for cognition. So when we are doing this interview right now, maybe both of us are using something like 5 watts. What is 5 watts? It's a flashlight. It's the power for a simple flashlight. And that's what it's what I'm trying to say is incredibly efficient. And then people say, yeah, but computers are more powerful. They can win at chess with humans. The blue gene computer beat Kasparov at chess, grandmaster and all that. Well, wait a minute. But this blue gene is powered by 1.5 megawatts of energy, which is basically the needs for a small town. 30,000 people in a town would require that kind of energy for their households. So we're beating, or let's say competing with supercomputers on one task, playing chess or playing Jeopardy or something like this, and sometimes winning. But our energy consumption at the same time is 1 million times less. It's pretty impressive, isn't it?
Incredible.
So how does it happen? And I'll go one step further. We have 100 billion neurons, and these neurons are active electrically, of course. So that's another important things to keep in mind. This is all electricity running through the neuronal networks in the brain. So if you divide 100, this number of 5 watts or something like that, flashlight by 100 billion neurons, so on average, each neuron consumes something like a picowatt. It's a tiny amount of energy. So now, why am I saying this? Because this electromagnetic energy of infrared light, the various devices provide, actually corresponds to some significant percentage of the neuron's energy demand. So that makes sense. It's not, airy-fairy. It is actually significant. Another, I want to bring in another device or therapeutic procedure, which has been used for about a decade now in the treatment of brain cancer. And that's electric fields called tumor treating fields, TT fields, produced by a company from Haifa, Israel called Novocure. and it's on the market. It's FDA approved, and I've worked with this company for a few years. We did experiments, and this is based on oscillating electric fields, low intensity. It's electric fields of the strength 5 volts per centimeter. So you can actually compare it to, again, a battery across a centimeter. The current flowing from the battery across 1 centimeter of some material, and they have very good clinical results for brain cancer patients who wear a helmet with these electrodes and the oscillating electric field at a frequency of about 100 kilohertz for 18 hours a day for the duration of their life, I guess. But the lifespan of these patients is typically increased by 50% or so. With glioblastoma, the prognosis is terrible, right? It's like 12 months.
The glial cells, right?
Yeah.
Super old. The bridge between the nervous system and
Exactly.
the immune system. If glial cells and the glial system is off, again, inflammation and, cancer.
So that's another energy. What's the term for it? Energy repairing technology for biological systems. You can call it electromagnetic energy that heals.
Well, and then also ultrasound and sound is being used to treat cancer as well, with sound being a waveform. Sound, because that's another treatment, right?
Yes.
Sound.
Especially, I think, ultrasound, so high frequency sound, as well as I'm not familiar, firsthand with this. I know, for example, Stuart Hameroff at the University of Arizona has developed some techniques with ultrasound being used for the treatment of neurological disorders, I think. But yeah, the principle is similar. I think it's delivery of energy, whether it's mechanical energy or electrical energy or electromagnetic energy, to augment the biochemical energy that mitochondria and glycolysis provide. So yes, I think energy medicine, which I know has somewhat controversial perception in the general public, like energy. But as a physicist, I can tell you, for me, it's not controversial. It just makes eminent sense.
It's like the universe, right? Energy, matter. It's like, hello, I don't know how energy's become a bad word in body treatments. It's like, that's what the body does.
Well,
The
exactly.
body is.
Exactly. And, so as a physicist, when I teach biophysics, I always tell my students that there are several distinguishing features of living systems as opposed to inanimate matter, one of which is metabolism. So energy transduction for the purpose of maintaining life we call it a metastable state, a state which is a non-equilibrium, thermodynamically non-equilibrium state, a higher energy state. And as we age, by the way, the height of this energy plateau decreases, and you can, from the physics point of view, you can say that the terminal point of life is when metabolism stops and the energy is at the lowest point.
Yeah.
So if you think about energy in these terms, then suddenly energy medicine makes eminent sense.
Yeah. Have you heard of the cymoscope? The
No.
cymoscope is the physics behind it is Faraday waves. So it's a technology that has that can take a snapshot of of where sound enters the medium of water, so that you can get a precise readout, a kind of sound signature, if you will, of the sound as it's imprinted on water. Now they're using cymoscopes to diagnose cancer, because cancer has a different sound signature.
It's not surprising to me. By the way, speaking of water, I mentioned earlier when I talked about the structure of the cell and 70% water, and that the water molecules in cells are not the same as in organized in the same way as tap water. And there is a body of work by Professor Gerald Pollack from University of Washington. You may also want to interview him if you haven't yet. A colleague of mine, and he demonstrated through his research and wrote several books and has fantastic YouTube videos showing how that water organization is different from running water in terms of it's almost like an ice structure organized in layers, which are not randomly oriented, but organized. And he calls it easy water. It's called easy. So letter E and letter Z or Z.
Oh, the fifth phase of water, right?
Yes, exactly.
Yep.
Exactly.
Yep.
Expulsion zone, or so this water forms a layer around proteins or hydrophilic surfaces. Proteins have mainly hydrophilic surfaces which like water, and this layer screens out some ions, so that can be over large distances, as long as micrometers, which will be spanning the entire cell. So according to Jared Pollack, most of the water molecules in the cell are coordinated in some way. So now we are talking about disease. And by the way, there's one more interesting thing. He demonstrated that this water organization is maintained by, or can be disrupted by, the absence of light. So when there's an experiment he talks about in his lectures that his student performed in darkness, and suddenly this layer became very short or shrank, and when you turn on the light, it grows. What's the meaning of this? The meaning of it is that light or electromagnetic energy is used by water molecules in cells for creating a different phase. So, symmetry breaking. So living normal, healthy, state of water is organized. Pathological states would have disorganized or broken symmetry states, and you restore it by providing additional energy. Could be electromagnetic. That's another aspect that I'm sure is taking place. So, and we talk about recharging our batteries when we go on holidays, and what do? We sunbathe, right? So we deliver electromagnetic energy through the skin to the rest of the body, and probably organize among other things. Okay, so
yeah, serotonin has a deep relationship with light in terms of evolution, and serotonin absolutely has everything to do with symmetry breaking in the embryo.
exactly.
Very interesting. It's kind of a side point here.
Well, it's not quite a side point, by the way, because if you want, we can follow along this line toward consciousness. Or you want
Okay,
to
yeah, let's go there. And then I want to come back to structural scaffolding and Ingber's work.
Okay.
Let's go there, serotonin, light, and consciousness. And then maybe we'll come back to calcium ions,
Yeah.
which is also probably related to consciousness.
Indeed.
Yeah.
Right. So what's the tangent on which I want to go here? The tangent is related to tryptophans. Tryptophan is an amino acid, one of 20 natural occurring amino acids that proteins use for their structure. Right? So tryptophan is special. It's the only amino acid that actually can be excited by electromagnetic waves near the visible range. Preferentially, it's UV. It's about 300 nanometer wavelength. Visible starts from 400, so it's a bit lower wavelength, but not much. And with my colleagues at Princeton, Greg Scholes, he's a well-known chemist at Princeton University. He participated in the explosion of interest in quantum biology, initially focusing on photosynthesis in plants and explaining and showing by experiments that this is a process that has a quantum mechanical mechanism at the bottom of it. So with him and my student Arat Kalra, who is now back in India, did the experiments at Princeton, we wanted to see if microtubules specifically are quantum operating in the quantum level, and that's again the connection to neurons, because microtubules and neurons are oriented in the spiral bundle and are instrumental in information processing and all these activities, transport, and so on. But importantly, there is a structural similarity in the tryptophan networks in tubulin, and tubulin makes microtubules, to photosynthetic systems. So in leaves, chlorophyll, when you look at the organization of specific aromatic groups called FMO, these are the light harvesting groups in leaves. And when you look at their distances and the architecture, it's very similar to tryptophans in tubulin. Okay. So that's the serotonin and tryptophan are very similar chemically. And there are some other molecules that have this indole group, specific chemical structure. Okay. So we did these experiments and excited tryptophan networks in microtubules and tubulin with laser light, and measured how long this light was re-emitted from these networks, from tubulin and microtubules, in order to establish the lifetime of quantum states, because these are quantum excitations, electronic transitions taking place. And we published this, and it's a very, I would say, nice paper, because it's accurate and has some very profound results in it. So we found that these quantum states exist for about 5 nanoseconds, up to 5 nanoseconds, which is short, but longer than in photosynthesis. So it may or may not be physiologically important. It's longer than leaves need for the generation of energy pathways, and actually they use it for glucose creation with light, water, and CO2, carbon dioxide. These are the ingredients for energy production in plants and some bacteria. Okay, so tryptophans in microtubules, 5 nanoseconds longer than photosynthesis, but not really. It's not bridging the gap to physiological processes such as occurring in our neurons. We need milliseconds at least, right? So ion channels, we'll come back. Ion channels, milliseconds. These processes, nanoseconds. So what's a nanosecond? Well, a nanosecond is a million times shorter period than a millisecond, 10 to the minus 9 seconds. So we have a problem, okay? It doesn't mean that it's useless, but it's a bit of a problem. And then the second thing that we were able to show is that these quantum states, they exist, but they are localized to about 6 nanometers, which is the size of one, roughly speaking, the size of one tubulin protein, one dimer. And again, it's a bit too small, because you want something that spans the length of a neuron, or at least a long microtubule, which is hundreds of micrometers long. So again, we have a problem with the scales. And that links to the, theory of consciousness by Penrose and Hameroff, 30 years of hundreds of papers on this topic. And that was the first experiment that actually delved deeply into this topic experimentally.
Yeah.
Which the answer is, a little bit yes, but I would say mainly no.
Well, this is what really got me going on your work. So you wrote a piece in Pathways to the Origin and Evolution of Meanings in the Universe. It's a book. It's an acronym, POEM. Pathways to the Origin and Evolution of Meanings in the Universe. And, because I had been teaching to students, I'm a neuroscience professor. I've been teaching to students the Penrose and Hameroff idea about quantum computation. And it made sense to me because I've been a microtubule guy for quite some time, and it made sense to me. But also, there is this scaling issue. I knew that I somehow knew that it's not quite all of it. And when I read your piece in Poem, I was just blown away. And because it just it I find it to be a better model, and your critique of Penrose and Hameroff is just brilliant, and the mathematics there. So it revolves around calcium ions. Maybe we can let calcium ions be an enigma for the listener for a little bit longer. Um, and I'd like to rewind. Is there anything else that you wanted to say about bioenergetics or serotonin? Or
Well, I could talk for hours about this, but I think in the interest of time, I would say let's move on.
Okay.
But we can talk about Penrose and Hameroff and ion channels next, because that's the natural segue.
Good. Okay. Well, I just want to come back just so that the listener can come back to kind of a base stance in the cell. And so, and because we were talking before so Jack talked about how the cell has this scaffolding, like this connective tissue.
Oh, yes.
So like, there's these filaments of various size. You have the microfilaments really small, the microtubules bigger, and intermediate in between them is the intermediate filament. So these are the guy wires that are really dynamic, that transport vesicles with, cargo like dopamine and so on and so forth. We've articulated mitochondria as being the fundamental to bioenergetics. Nothing's going to happen without mitochondria. And you also have, and this is just one cell, right? So microtubules we'll continue to talk about, but I want to talk about this very essential protein that has to do with relationship called integrins. And I wanted to just name integrins, and I wanted to name something that you've kind of been dancing around a little bit, this phenomenon called mechanotransduction. So I wanted to put those two pieces out, integrins, because that essentially links the cell to the entire collagen matrix of the body, correct?
Yes. Exactly. So it's a great introduction to this topic, and I want to build up on this. So when I started this conversation with the structure of the cell, we didn't talk about the mechanics of it all, just what's in it. But it's not pieces floating aimlessly without mechanical tension involved, and this topic has been beautifully described by Professor Donald Ingber from Harvard. He introduced the concept of tensegrity, which is a short for tensional integrity. That means that the integrity of the cell is not just by, let's say, containing all the components within the membrane, but by pre-stressing them. And he made a analogy to setting up a tent. If you go on a camping trip, you set up a tent, and you have the canvas, which is the membrane, but it would be floppy and not stable. So you have to put a a post, and that would be, let's say, microtubules, and then you have actin filaments, which would be stressing the different parts of the canvas, and then you put the stakes on the ground, which will be integrins, which attach the tent, the canvas of the tent, to the ground, and everything is under tension mechanically, which means that you have a stable structure. If you cut some of these pieces, the different posts and poles and well, then of course it can collapse. The tent would collapse, and that's sometimes needed when the cell, of course, divides. It cannot be so structurally tense because otherwise couldn't possibly divide into two parts, and then rearranges itself and so on and so forth. But that's exactly what's involved. Namely, there is mechanical interaction taking place between all these parts in a specific way. Of course, it's not random, and that forms the integrity of the entire cell. So I hope this kind of describes in very simple terms. So think about this tent, setting up a tent from parts. Each part itself doesn't have any specific well, they have specific roles, but only within the context of the tent, because the canvas itself is floppy. You can't really build any. You have to have a post, and you have to have the struts, and you have to have the cables, and pretense the different parts, like the actin filaments, which are used for this. Now, there's one more thing. So this is just mechanics, but many of these parts have incredibly intriguing and important electrical properties. I'll just talk about two. The first one is the membrane itself. The membrane itself this is a phospholipid bilayer, and has dipole moments, which are very strong. So the electric field between the two layers, these two phospholipid layers, is comparable to the electric field in the breakdown in strength when the lightning strikes. It's about 10 to the 7th, 10 million, on the order of 10 million volts per meter. So it's enormous field, right? Jeez. So now you have the electromechanical connection. And by the way, when this field is reduced, so typically the potential difference between the two layers is about 100 millivolts, 0.1 volt, small. But over a distance of 4 to 5 nanometers, that gives you this huge electric field intensity. When that potential drops to about 0.02, 20 millivolts, then you have pathological situations. And that's characteristic. One of the key characteristics of cancer cells is the reduction in the potential across the membrane of the cells. And by the way, this is another connection to your future speaker, hopefully Mike Levin from Tufts University, who has dedicated his professional career to understanding the importance of electric fields in things like cell differentiation, and morphology and so on. So let's keep this in mind. The membrane has an incredibly high potential difference, electric field intensity, and when that is reduced, it loses its functional usefulness, because cancer cells are not useful. They don't contribute to the normal cell physiology of tissues, right? So that's one of the hallmarks. And the second aspect is microtubules. So microtubules have one of the highest, if not the highest, charge densities, electric charge densities of all the proteins. They are an outlier, with about 50 charges, negative charges per building block per tubule and dimer. And that has consequences, which I spent the last, decade or so of my career trying to understand the consequences. One of which is conductivity, electric conductivity, ionic conductivity. So maybe, if you don't mind, I'll spend a few minutes about this, how that electric charge linked to the mechanical properties. Microtubules are the most rigid structures mechanically in cells, and they're also the most electrically active. So you have the connection, electromechanical connection.
Yeah.
So we measured conductivity, electric conductivity along microtubules, and we found this conductivity to be very high at low ionic concentrations, and quite low at high ionic concentrations. So it's tunable. And as you know, ionic concentrations of cell change, and calcium waves in particular regulate a lot of activities. And they, by the way, destabilize microtubules. Calcium makes them less rigid, more flexible. And that's the origin of mechanical waves that are used, for example, in cilia and flagella for propulsion, for movement, right? So this is the connection. Ionic flows affecting the mechanics of microtubules, and the mechanics of microtubules is used by the cell for movement, for example, in bacterial cells.
Oh, and in amoeba too, because amoeba send out their pseudopoids. I think they're called pseudopoids that have these microtubule, microfilament structural scaffolding to kind of pull them along.
Right.
Yeah.
Yes. So that's one thing, but there is more, and I'll just say a few words. I don't want to get into the weeds because there's a lot of technical stuff, but one thing is conductivity. So you can have, basically you can think about now the cytoskeleton, microtubules, actin filaments, as electrical circuits inside the cell, and they send signals. So we did measurements with the help of my colleague Horacio Cantiello, who was at Harvard at the time, and now he's in Buenos Aires, showing that actually microtubules can act as transistors. They have exactly the same properties as transistors. And then later on, we showed that they have some other intriguing properties, depending on the way you stimulate them electrically. They act as memristors, which is another interesting structure discovered by Leon Chua from Berkeley. We published a paper with Leon Chua on this. That means that they maintain the memory of the previous event, electrical conduction event. So they are transistors, memristors, conductors, and they also have capacitive properties. So they can store charges. And everything, the function depends on the conditions. So they are incredibly multifunctional structures. And then finally, with Aristide Dogario, whom I mentioned, I think in Florida, we showed that when they are exposed to visible light, so we use laser at 660 nanometer laser light, you can slow down the formation of microtubules and prevent some microtubules from forming. So light can be used also for interacting with these structures in terms of the dynamics. So all of this is, I would say, spots on a map of properties, and we still need to connect the dots. So we have these dots, but we're not completely there yet in terms of connectivity.
And connecting across scales, which is very challenging. So we've been talking about cells and cell architecture and microtubules and filaments and so on. And then this is why I like integrins, because they connect the cell to a cell sheet, like in epithelial cells, and plugs that cell into the whole mechanical tension grid that is the body as a whole. This is connective tissue. This is the body. And it's like, calcium waves and calcium ions are long range communication molecules, right? Long range communication. And so is the mechanical tension grid of the body as a whole. So, things like conscious breathing and stretching, yoga, and I'm a myofascial release practitioner. So I get in there and stretch the fascia. And that's got to be releasing energetic processes just by stretching, getting the mechanical tug going in the body.
Absolutely. I think there is that connection, and there is also the aspect of across the body communication between cells. Some of these processes can only be explained by electromagnetic interactions because there's just too too long a distance to cover in too short a time. And as we talk about the speed of interactions, the slowest, by the way, are chemical. Chemical interactions are very slow. Then slightly faster could be some diffusive processes, especially bias diffusion. And then you have electrical signals, which are faster yet, up to, let's say, 10 volts, 10 meters per second, which would be 36 kilometers an hour. But still doesn't explain everything. For example, our reaction times to when, look at, I play tennis. When you look at some of the best players in tennis and you have a serve or the ball going at more than 100 miles an hour, the reaction time is just impossible to explain otherwise than through super fast electromagnetic speeds. So electromagnetic is speed of light, right? So then you have 3 times 10 to the 8th meters per second. So you see, we have all these channels of communication: chemical, mechanical, electrical, and electromagnetic, and they serve different purposes. But we cannot simply say there's one.
Yeah, I mean, there's this fancy word that brings it all together that Dick Gordon, who I'll be talking to soon, in fact, right after this call, I'll be jumping on a call with Dick, who's a wonderful embryologist. I just love his work. He's a calcium ion guy as well. It is just, electromechanochemical.
Exactly.
Electromechanochemical. It's like it's a mouthful, but it fits it all. It's all right there, right? And the mechanical structure interfaces with the chemicals. Like there's some theories out there that the microfilaments go down and via link complexes get smaller and smaller and smaller and interface with the actual chromatin with the DNA structure. That there's some interesting articles about that, and then of course, expanding out to the body as a whole and the whole body systems. And so we're an integrated electromechanochemical whole, and yeah. So I'm wondering if we can, well, I'd like to kind of get a sense for how because, uh, Penrose and Hameroff, they had this quantum computation model of microtubules, and which they talked a lot about memory,
Mhm.
which you've done as well too. But to me, I think you've updated the model, and you're really the bridge into the future of quantum computation, because I'm with you. I think calcium ions are a better contender. Can you tell us how you came about that insight and
Yeah.
what's up with that?
Yes. 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 Okay. So it's a little bit detailed, but let me try to simplify it. Um, so I talked a little bit about these experiments at Princeton with the time scale and the spatial dimensions of quantum states. We we are more or less in agreement that there are quantum states involving microtubules, or there can be. Uh, what I have difficulty with is three things actually. The first one is uh, the the short period over which these quantum states are maintained. So maybe cells, neurons in particular, found a way of elongating them by a million times. It's a tall order, to be quite honest. The second thing is, how do you expand them, create this coherent domain called coherence, quantum coherence, right? Coherent domain over the entire microtubule. It's already a challenge. Maybe you can do it somehow um, with making them parallel and interconnected. Okay, perhaps. That's the second challenge. And the third challenge is energy. Where the heck do you get this energy? Because in this particular experiment, the energy was in the ultraviolet. And for all I know, there is very little you can talk about in terms of sources of UV energy in living systems. Why is that? UV energy comes from the sun, right? So that's why we use sunblock and so on, and polarized glasses to avoid damage. UV energy is damaging to DNA, breaks DNA. So if that was the mechanism for fueling computational processes in the brain, that would have tremendous risks, right? Let alone finding the sources for it, because it's high energy. And we talked about ATP, ATP being the molecule of quantum biological energy. I can give you numbers. It's about 0.6. It has the energy of about in joules, at about, if I remember correctly, 0.6 joules. Sorry, no, 0.6 electron volts. Okay, so UV light is about 4 electron volts. So you cannot easily imagine a process which would, utilize, let's say, 6 or 7 ATP simultaneously molecules to get to that element, to that level of energy. People talk about the reactive oxygen species recombining, and yes, they can produce a little bit of UV. But our brain would not be effectively functioning with this finicky process of stochastic recombination of some molecules, right, providing the fuel. We need to have, as we know from other examples in cell biology, everything is well organized, orchestrated, and on demand, appearing just in time. So if we were to, and obviously if we were also going further, if you had too much or too little of this reactive oxygen species production, then your mental processes will be affected. And I don't know of any evidence for this. So the energy crisis here is profound, I would say. And that leads me to your question about ion channels, which is a better candidate. There's a second argument, namely power. I talked maybe an hour ago or something like this about the power consumption, right? 25 watts per brain, picowatts per neuron.
Yeah, we're using about 5 watts now. No, I think I'm up at about 7 or 8, Jack.
We're improving. So when you do this calculation, and this paper that you mentioned in Poem, I presented the calculation in detail. It turns out that power is energy divided by time. So if you have a short amount of time, you for a given amount of energy, that requires a lot of power. And I did this calculation for tubulin, quantum states and tubulin, and the conclusion was that there was not enough power in the neuron, even if all of the energy was used for computation, to process information per tubulin unit. Only the entire microtubule could be in one quantum state. And then you could explain it. But we don't have any evidence for this. In fact, not only one microtubule, but the entire neuron could be only one state, quantum state, or the other quantum state, given the rate of transitions. So the hypothesis that Penrose and Hammer have proposed talks about tubulins being in qubits, quantum states, right? Qubits for information processing. And that cannot be reconciled with the amount of power used by neurons, because it's too many units. I don't remember now, 100,000 to 1,000,000 dimers in a neuron, and they all change states on a nanosecond time scale. Then you get an energy catastrophe, basically. You would blow up the brain with the amount of energy needed for this. However, when you look at ion channels, I did this calculation. Ion channels operate on a millisecond time scale, and there is a smaller number, maybe 1,000 or so ion channels. then everything works well. So ion channel functioning as a let's say qubit is consistent with the amount of energy that we know the brain uses. And furthermore, there's work done by, among others, Gustav Bernreuter from University of Salzburg in Austria, and Vahid Salari, who is now in Calgary, not far from here. They wrote a number of papers showing that ion channels operate on quantum principles. Ions traverse the channel as a quantum tunneling process. They have a wave function which basically propagates through the ion channel in a quantum tunneling way. So that to me is much more believable, although we are still hypothesizing. But I would say that has a much higher probability than microtubules, doing all this. However, and you mentioned the memory. However, they can be used for something else, and we know that they're used for many, many things: electrical signaling, mechanical stability, even regulation of ion channels. You are aware of the fact that microtubules interact with voltage-dependent ion channels and anion channels, and they can modify their activity, but especially by C-termini, these protrusions which are highly charged. They can stimulate or block and whatever. So,
and what's true in the kind of adult body is very, very true in the construction of the body and embryology as well, which microtubules and calcium ions are central. Calcium waves are central to actually building the body as well.
So you see, now we are connecting the dots, because although microtubules may not be the principal modes of quantum processing, information processing, but they may be regulating the functioning of ion channels, which may be those central to information processing in the brain. And signals can be further propagated through microtubules into the cell and processed. And that is, by the way, also something that was demonstrated by the group of Donald Ingber we talked about earlier. Back in the 1990s, his then, I think, research associate Andy Maniotis and Donald Ingber published a paper, I think it was in Science, showing that microtubules transfer mechanical information or signals across the cell into the nucleus. So now you see the connectivity of the cell. We talked about not only mechanical tensegrity, but also information processing from the membrane, regulating channels, sending signals across the cell into the nucleus, maybe triggering transcription factor activity and then production of proteins on demand that may be needed for something. Let's say, okay, let's start reorganizing the cytoskeleton and change the orientation or go into cell division or things like that. So I think that's where I think the integration of information and signals, mechanical, electrical, chemical, comes into play.
And then tell us a little bit more about memory.
Right. And this is our hypothesis, which goes back to our paper in 2012, by the way, with Stuart Hameroff and my former student, Travis Craddock, who is now professor at the University of Waterloo. So we wrote a paper based on some experiments that were performed at, I believe, NIH, maybe 2011, where it was discovered that inhibitors of a particular enzyme, calmodulin kinase 2, erases memories in experimental mice. So the mice in these experiments were solving some mazes, typical experiments, and they remembered all of that. And then they were given this inhibitor. I don't remember the name of the pharmacological agent, and they forgot everything. And this enzyme calmodulin kinase 2 or CAMK2, we looked at it, and what it does is phosphorylate microtubules in neurons. So phosphorylation, as you and your audience may know, is a signaling process in biology, very typical. So activation of some sites on proteins, typically by phosphorylation. So that enzyme phosphorylates, in this particular case, serine residues on C-termini of microtubules. And we looked at
Serine, as in D-serine?
yes,
Interesting.
yes.
They're also implicated in TBI, by the way.
Oh,
Serine.
interesting.
Deep.
There you go. That's
Yeah.
I didn't know about this connection, but that's exactly. And we created computer models showing how phosphorylation of microtubules by this enzyme is consistent with the structural compatibility of the two. And this paper shows exactly how it fits into the structure of microtubules, and also electrostatically attracts the two attract each other because of the charge complementarity. And then we also went further and we calculated the amount of memory storage that can be implemented across the brain with this single process. And it's mind-boggling. You can actually encode the entire Library of Congress just by using this as bits of information, right? Phosphorylation on different dimers, staggering numbers. So the entire memory of your entire lifetime, all the inputs can be easily stored by this kind of information encoding. So phosphorylation, let's say, think about it, 1, and dephosphorylation is 0. So you have two possibilities, and you have so many sites.
Now, is there overlap? There is a little bit of overlap with Hameroff and Penrose's theory of memory acquisition via microtubules. Am I right in saying that? How's it different?
It's a little bit different. I would say that this one is this mechanism I just described is based on molecular biology, and it is consistent with. It's of course slower because you need to have this enzyme, which it's like a motor protein, moves at the speed of, let's say, 1 micron per second, up to maybe less, maximum. So and the microtubule is 20 microns long, so you walk through it over 20 seconds. So it takes minutes to encode all of these things. And
because it involves chemical phosphorylation is a chemical process, the slowest process. Yeah,
exactly.
Yeah. Okay. Well, that makes a lot of sense. So, and then I was also thinking, there in neuroscience, there's this very common phrase that's bandied about called when neurons fire together, they wire together. Now, fair enough, and there's microtubules have got to be involved in this process, but there's excessive focus on the wiring. And, in my book, I offer an alternative phrase, and it's kind of a catchphrase, which is when cilia sway, memories stay. Because it's been shown that, the hippocampus is, central to memory formation. And what one research finding found is that it's actually cilia that are central to memory production. And it's quite interesting, right? Because, cilia, the structure of cilia is held together by microtubules.
Exactly.
And cilia are a perfect bridge between, or I would say the glue, right, between electromechanical.
Indeed.
Yeah.
Yes. I think it's so we're on the quest to connect the dots. I didn't know about this property of cilia and memory formation, but it makes a lot of sense to me. I have to read your book.
Very cool, and I can send you that article as well. There's two other big links. One is embryology, which cilia are central to. There's the discussion about broken symmetry, but and then there's also evolution, which I just wanted to note to the listener that in origin of life theory, of course, there's at least 24 different theories about the origin of life, and it's not like it's a done deal at all. But one of them is called the vesicle first hypothesis, essentially the bubble first hypothesis, that
Yeah.
before there was life, there needed to be a bubble. And, one of the theories is that the bubble, once it started to gather lipids around it, needed to be resilient enough to hold calcium ions. Yeah. You with
Yeah.
me? So what
Yeah.
what are the implications of that? This origin of life hypothesis, that it has something to do with trapping calcium ions inside a bubble that doesn't break, because actually lipids that hold the bubble together, calcium is toxic to those lipids. So pre-life needed to figure out how to hold the bubble together and the calcium inside without popping. What are the implications of this hypothesis?
Hmm. I don't know, but I can tell you that microtubules emerge, a tubule in fact emerged a bit later because it's only present in eukaryotic cells. Prokaryotic cells have an analog of tubulin called FTSZ, but it's not forming such structures. It's forming rings rather than cylinders. So there are some steps to again in the evolution that need to be taken to connect again these two processes to structure.
Well, calcium ions are, you know, kind of like your contender for quantum computation. And I think the math is there. It really lines up in this poem book. I think you just did a wonderful job.
Thank you.
And but also calcium is central for calcium waves a la Dick Gordon,
Yeah.
central to creating bodies embryologically. It's central to long range cellular communication. And Michael Levin says that cancer is an error of geometry, so that the structural scaffolding is off, and then potassium and calcium ions, the long range communication gets bent out of shape. So, and these are hard dots to connect. Like calcium has a very, very ancient
Mm.
presence all the way back into pre-life. Um, and now you're saying, well, calcium ions are the best contender for a quantum model of biology.
Yes.
And
Yes.
in between somewhere is how the embryo holds itself together, and you know what I mean?
Yeah.
And that has to do with the question of cancer.
Right. And by the way, confinement, so forming a finite vessel in which life can thrive, exist, be initiated, also makes sense from the point of view of um, water, structured water. So the medium in which all of this exists needs to be organized. If you have an open system without boundaries, water is not formed like that. Confinement. And there was a group in Japan I visited a few years ago. They measured the smallest size of a container, vesicle that would be conducive to water organization in a structure like a la Jerry Pollack. And it's something like I don't remember now, maybe 10 micrometers across or something. They determined the maximum size. So there is something there as well. So the medium needs to be of special type. Otherwise, you'll have decoherence, which is not conducive to or synchronization of activities and all that. At any rate, that's my.
Well, okay, so I'm wondering if we could kind of orient to completion with maybe the most ambitious part of the discussion, which is because I do believe I remember reading in your work something about pilot wave theory, uh, calcium ions and pilot wave theory. So maybe we could kind of go way out into the cosmos for a moment and talk about, biophysics and how it interfaces with astrophysics?
Oh, I see. Well, I guess no. I have to say I've not worked on the pilot wave theory. It's somebody.
Okay, so I got it bent out of shape.
No, it's not not my area of expertise, to be quite honest. I can talk about quantum, but not pilot wave. That's Bohmian mechanics, I think. Paavo Pilkkanen, if you want to um, interview him, he's in Sweden and Finland. He's actually a student of David Bohm, I believe, and talks about this in connection with consciousness and cosmic consciousness, things of this nature. So I have to apologize, but I'm kind of limited in my scope of knowledge to things on a smaller scale.
Oh, okay. So it's me that got it wrong. What's this guy's name?
Paavo Pilkkanen. Paavo
Pilkkanen.
Pil-
Pilkkanen. I might have run into his name before. Well, I mean, so clearly there's waveforms in the universe, particles and waves. And, there's waveforms in the body. Calcium ions fluctuate in waves, certainly embryologically in building bodies. But do you know anything about wave fluctuations in the cell and in the body as a whole? Like ion fluctuations, wave-like fluctuations in the body?
Not much, but what I want to say is this: that it all boils down to synchronization of activity. So this, what Stuart Hammer was talking about as the unitary sense of self, that all the cells communicate within the body, and how it happens, we don't know, but it's probably some form of quantum entanglement, right? The Nobel Prize in Physics 2022, quantum entanglement. We now know that this is for real. Somehow Einstein never bought into it. He said that this is the spooky action at a distance, but now we know it's a fact that quantum particles can get entangled over up to infinite distances, photons and electrons and so on. So, of course, we don't have any proof that this happens at the level of biological systems, but it would be incredibly useful to involve in cell-cell communication, involve in entanglement throughout the entire body of all cells, in under normal conditions, disentanglement or some kind of phase transitions from quantum to classical. In pathological states, maybe different organs or different tissues would be decoupled, and then you have pathologies. And then you're going to go beyond. You can talk about entanglement between humans, right? If we as entities are human entirely and are entangled internally, we can also imagine easily that we can get entangled with other humans. And then you have this sort of planetary coherence, right?
Well, I love Dick Gordon's reframe on evolution, that it's not a tree, that it's a tangled bush of entanglement. I think that's a cool reframe.
Yes, and in fact, evolution itself could have been easily understood as accelerated by entanglement, by quantum search algorithms. Otherwise, they're very difficult concepts to explain, all the variations in species and optimization of adjustment to environmental niches and so on. So by brute force computing these possibilities, you end up with an impossibility. If you take it classically, but quantum mechanically, everything changes, right?
Lowenstein does a great job of kind of unpacking the big picture. Did you ever read any Lowenstein?
Yes, yes. I'm a big fan of his work.
Oh, it's such a great, the foundation stone, I think it's called, the foundation stone of life or something.
Touchstone of life.
Touchstone of life. Touchstone of life. That's brilliant. I just love that stuff. I'm wondering if, as a final comment, you can give the audience, because you're a cancer researcher, well-renowned cancer researcher, your current hypothesis about what's happening with cancer.
Ah. Well, again, this is a huge topic, and I don't want to, you know,
Well, yeah, reducing complexity, we're just like,
that-
it's like going up to 9 watts.
but I think, in the simplest terms, um, well, it's a phase transition. I wrote the paper, cancer is a phase transition. It's a broken symmetry state from in physical terms, but more specifically, it means that cancer cells um, are well, either through energy crisis, for example, an energy crisis, lack of oxygen, hypoxia, or lack of fuel, they switch to a different mode of operation, namely from this normal oxidative phosphorylation, metabolic transition to glycolysis, inefficient and with all the toxicity related to it. And so that's, to me, the most the simplest and the most powerful explanation. It goes back to Otto von Warburg, Nobel Prize winner in chemistry, 1926, formulated this hypothesis called the Warburg effect, Warburg in German, um, and that is the initiation of the process. Then you have a cascade of events, multi-step process, which is also demonstrated in the epidemiology of cancer and so on, that there are subsequent um, genomic instabilities occurring. So it probably starts with a metabolic instability, a lack of metabolic energy for various reasons. Could be, mechanistic. It could be lack of oxygen, as I mentioned, and so on. And then there's concomitant electric changes, which I mentioned, with membrane depolarization of the membrane, leading to imbalances in ionic concentrations, you back to calcium and sodium and potassium, as well as pH, which is also demonstrated abundantly. So acidic production of the acidic environment compared to normal cells, and a basic insight. So changes in pH up to a 0.5, 0.6, which leads in a positive feedback loop to the shutting down of mitochondria, because the mitochondrial energy production rate is proportional to the pH difference between the cytoplasm and the mitochondria. So if pH in the cytoplasm increases because it's becoming more basic, and mitochondrial pH is something like 8.2 or 8.1, in normal cells, in cytoplasmic, let's say 7.2, it goes up to 7.56. You suddenly have a lower potential difference, and the rate of energy production decreases. So that's kind of maybe not a simple explanation, but it's a multi-step.
And it's physics, right? You're talking symmetry. Symmetry, one great researcher once said that, physics is the study of symmetry and how it gets broken. And what you're saying, what I'm hearing you say, is that this is a problem of physics, that there's a phase transition of broken symmetry.
Exactly.
Yeah.
And that leads to, subsequent decoupling from the rest of the surrounding tissue and so on. So that's how I see it. You mentioned in the beginning that I worked on oncology from the pharmacological point of view, drug design, computational drug design. I still do, in fact, but I'm now more excited about other means of cancer therapies, not just pharmacological. I'm not dismissing it, but it's a very slow, gradual, and expensive process. If we could find electromagnetic or other, physical therapeutic modalities that overcome the problem of toxicity that is related with cancer chemotherapy, that to me is the future prospect that I would really want to see.
That's really exciting. Like things like infrared and sound, where you have interface with the body as a field, make it more coherent.
All right. So I I hope that I get some people interested in a few topics that we discussed.
Yeah, thank you so much, Dr. Tuszynski. It's been really fun, and thank you so much for your brilliant mind and your mathematics and your wonderful theories, and I hope to talk again.